Bioinks are hybrid materials made by two main components. One is cells that can be any kind of cells, and the other is a polymeric structure. And cells and polymeric structure are usually mixed before to be printed to construct a three-dimensional model. In our project, we'll test different biomaterials, three different polymers, to find the best one in terms of bioavailability, and we will select the biomaterials and the bioinks that will be the best one to reconstruct the urethra. Urethra is a tubular structure that starts from bladder. For several reasons, after surgery or after a traumatic event, can be damaged. So this kind of problem in this project will be fixed by 3D bioprinting. In other words, we want to try to reconstruct by using these bioinks this tubular structure. In this project, we will have two different approaches. One for simple damage, so when there is really a local damage, and we will do something that we call the fast procedure. So we will take cells from the animals, the best biomaterials that will be selected during the project, to reconstruct by 3D bioprinting immediately these little fissures, this little hole. If there is a huge damage, so we need to have a long procedure, we will also replicate the whole cylindrical structure, all the tubular structure. In this case, there are different stages that are required. So we will collect the cells, put the cells in a bioreactor, reconstruct in laboratory the whole cylindrical structure, and after, in the second moment, we will replace this structure in the animal. So we will want to develop two different protocols, one more focused in a very little damage, can be fixed very easily, or another for a huge surgery reconstruction of the entire urethra.